For centuries, the prevailing view treated thoughts as immaterial and abstract, processes existing independently of physical biology. Neuroimaging has shifted that perspective by making the physical footprint of thought visible to clinical observation. Comparing these two datasets reveals nearly identical thermal signatures localized in the primary motor regions. Dataset A reflects the brain activity of a subject physically performing a complex piano sonata. In dataset B, the subject is perfectly still, but they are vividly imagining playing that same musical sequence. The activation patterns in the primary motor cortex remain virtually indistinguishable across both conditions. This is captured through the bold signal, which tracks oxygenated blood flow as it is diverted toward neural tissue in active use. The presence of the bold signal in the motionless subject proves the motor cortex maintains a measurable metabolic demand strictly through the internal act of imagination. Based on this evidence, a thought is not a purely abstract event. It is a physical act occurring within the microcosm of the brain. To understand exactly how a non-physical thought drives this metabolic demand, we must look at where this activity is localized. The activity is centered in the precentral gyrus, the primary motor cortex responsible for executing voluntary movement. Imagining the sonata bypasses the general thinking centers of the prefrontal cortex, instead activating the specific topographical region assigned to the hands and fingers. The brain recruits the precise biological circuitry required to move the body, even without mechanical output. A subtractive analysis shows a near-total topographical overlap. The neural hardware used for imagination is the exact same hardware used for action. This relationship is known as functional isomorphism. It describes a state where the neural architecture used to simulate a movement is completely identical to the architecture used to execute it. The motor cortex does not distinguish between a physical performance and a sufficiently vivid simulation. For the brain's motor centers, actually touching the keys of a piano is strictly unnecessary for the system to reach full activation. Because functional isomorphism links imagination to physical hardware, we must examine the biological consequences that follow at the cellular level. Shifting our view from macro-level scans down to the microscopic cellular environment, we see the direct results of this activation. Every bold signal represents millions of individual synapses firing in sequence. This occurs at the synaptic cleft, the physical gap between the presynaptic terminal sending a signal and the postsynaptic terminal receiving it. An imagined thought produces a chemical action potential that must cross this physical gap. This process is dictated by the principle of Hebbian plasticity. The governing rule is that neurons that fire together wire together. When a thought activates these specific neurons, it initiates a localized chemical cascade. This cascade functions as a structural reinforcement mechanism for the active pathway. The synapse undergoes physical change. Receptor density increases, and the connection strengthens because the simulated command required it to fire. Only the pathways used to simulate the piano sonata receive this biological upgrade. The surrounding inactive tissue remains exactly the same. These fortified pathways become more efficient. Internal thought provides a biological trigger sufficient to physically rewire synaptic connections within the motor cortex. Over weeks of mental rehearsal, these microscopic synaptic changes compound, leading to measurable macrostructural changes. Longitudinal imaging allows us to track permanent changes in the brain's white matter tracts. White matter tracts are bundles of axons providing the structural connectivity between different processing centers. Using diffusion tensor imaging, or DTI, researchers measure the physical thickening of these pathways. The data shows that repeated mental imagery causes these tracts to expand in diameter. The outer myelin sheath thickens, providing better insulation and increasing the speed at which electrical signals travel through the tract. This creates a fortified biological architecture that differs significantly from an untrained brain. The data reveals that the motor cortex has undergone structural optimization for an action the body has not yet performed. The internal thought act is the primary driver of this process, setting the biological stage for all future physical performance. By examining the physical overlap between action and imagination, we can see how thought triggers genuine synaptic firing. This internal simulation mirrors physical execution with enough precision to engage the brain's primary motor hardware. This engagement is not temporary. It leaves behind a measurable cellular footprint through Hebbian plasticity. As these pathways are reinforced through repetition, they lead to long-term structural changes in the brain's white matter. Because thought produces the same metabolic and structural changes as physical experience, it functions as the primary internal mechanism for rewiring the brain. By physically altering the instrument through which we perceive and act, the thought act does more than process information. It continuously remodels the neural architecture of the self.